details of the arrangement of the proteins that build the anatomy of, after we're done with this, you might say to yourself now, I know that he mentioned smooth muscle and cardiac muscle also. So, isn't that sort of conspicuously being left out here? Especially since I distinctly remember him saying something about the different arrangement of actin and myosin in smooth muscle. Surely he's going to say more. Or maybe you say to yourself, I know he mentioned the similarities between skeletal and cardiac muscle, but he did not finish this introductory material by talking about specific differences in the anatomy of skeletal and cardiac, which I know there are some. So I'm well aware of those questions that will come to your mind, and so we'll just put that to rest here. The cardiac muscle will in fact be discussed. We're going to spend a whole section on heart anatomy in this class, so that'll come up. And in the smooth muscle story, we'll resurface again whenever we talk about respiration and digestion. There's certainly more to be said, and I could choose to do the urogenital system with that, but that comes last in the semester. And so when we get to respiration, the smooth muscle story will come up again. And at that point, we'll talk about the incredible abilities of smooth muscle to stretch, and that'll carry right on through the end of the semester. So I haven't forgotten that there are two other kinds of muscle, but the first portion of the course after this introductory material is all about skeletal muscle. And so that's why we finished the introductory material with it. Okay. So last time we talked in sort of overview terminology about motor neurons and somatic fibers and autonomic fibers. And we had made our way this far in the motor pathways. Remember, you should put these sequences together in your mind. They will become increasingly important for you. We start in the precentral gyrus, and then we ride that axon down the lateral funiculus, all the way to the ventral horn of the appropriate spinal cord segment. We leave the spinal cord on the ventral side, and for the upper and lower extremities, we become part of a net of motor outputs, plexuses we call them. And then we can track that nerve all the way out to the muscle, remembering all the while that nerve has information going the other way as well. So we've made our way all the way out by way of the motor projection here to the skeletal muscle fiber. And you can see the ends of this motor neuron communicating with the skeletal muscle fiber. Now, let's point this out first. Terminology. When you say muscle fiber, you're talking about skeletal muscle. What you mean is the muscle cell. So this would be an individual muscle cell. Here, you would find. Let me just give you an example of the magnitude of this, because this is a very small picture. If you had, for example, you could shrink yourself down onto Miss Frizzle's bus, and you could jump aboard the bicep muscle here, and you got to a single cell, and you were on the membrane, on the sarcolemma of a single cell. Way down here next to the radial tuberosity, the attachment for both heads of the bicep. And you could ride that cell all the way up this direction, and it would either go to one of two places. It would come up here and attach to the rim of the glenoid cavity, or it would come across this way and attach to the coracoid process. If you were to do that, and you count it like you're riding on a highway, across the bridge, you would be crossing these striated patterns here, these places that we're going to identify as sarcomeres And as you did that, you would cross something in the neighborhood of this muscle cell, from here to here, about 100,000 of these sarcomeres. So we're looking at what? For the anatomy here, 1, 2, 3, 4, 5, 6, 7, 8, 9. So it gives you an indication about how small we are here in terms of talking about the muscle anatomy. Okay, so the motor neuron comes here and it connects with the muscle fiber, one cell. That's what we mean by fiber. And it does so in a very specific way. This junction is a very famous junction between motor neurons and skeletal muscle cells called the neuromuscular junction. You can see the end of the axon here. Looks like it's plugged into the sarcolemma, but it's not touching. There, in fact, is a gap here between the end of the motor neuron and the sarcolemma called the neuromuscular junction. So, I will spend a great deal of time this spring talking with you about the implications of all of these various pieces and parts that actually make the muscle contract. So you have something to look forward to later in your lives. For now, I want to point out one thing that will allow me to go back to my all-famous picture one more time. I want you to notice that whenever this signal gets to the end of this motor neuron and it's going to communicate with the sarcolemma, it's going to do so by using a molecule that is arguably the most famous of all of the neurochemistry, muscle physiology molecules in the human body. Is this one, abbreviated ACH. That is literally the word acetylcholine. Now some of you recognize that word because when we were doing this on Friday, we talked about the somatic and the autonomic pathways. Some of you noticed this. When you looked at this, you said, I wonder why he's not talking about that. Do you see it? So you see what I mean by acetylcholine being important? If I'm going to talk about how the nervous system controls skeletal muscle, I have to talk about acetylcholine. So the physiology of that will become incredibly important. But that's not the end of the story for this neurotransmitter because look, if I want to talk about the preganglionic sympathetic or the preganglionic parasympathetic projections, you see it? I have to talk about acetylcholine. So whether I'm talking about the voluntary nervous system or the involuntary nervous system, the story is about acetylcholine. The somatic fibers released it, the preganglionic sympathetics release it, the preganglionic parasympathetics release it. Now, the only thing on this picture that stands apart from that is the postganglionic neurotransmitter of the sympathetic nervous system. So here's your postganglionic projection releasing norepinephrine. The rest of it is the story of acetylcholine. Okay, so that gives me an opportunity to bring that up. So at this point, I have joined the motor neuron with the sarcolemma, and I have communicated. All you know at this point is the acetylcholine story. And I've used some terminology. I've used muscle fiber, I've used neuromuscular junction, and I've used the word sarcolemma, the membrane of the muscle cell. Now, if we were to break this muscle down, any individual muscle down, we would do so kind of like we did the neuron. Sorry, kind of like we did the nerve. And so the epimysium covers the entire muscle group. If you had to guess what the individual parts of the entire muscle were, the bundles that actually build the muscle, or if we were to talk about the bundles that build the nerve, we would call them fascicles. The bundles that build the skeletal muscle are called fascicles. The bundles of axons that are used to build the nerve are called fascicles. The whole nerve is surrounded by epineurium. The whole muscle is surrounded by epimysium. The fascicles are surrounded by In the muscle, paramecium. The fascicles are surrounded by, in the nerve, perineurium. Okay? And then finally, as we pull the muscle cell, our muscle fiber, see how it's in parentheses there, we pull the muscle fiber out or cell out. This was, in fact, in the neuron, the axon. Yes? But in the muscle, it is the individual cell. We could have said that here, right? That's an axon of one cell. So here in the muscle, it's an individual cell, and those cylinders, all those cells collectively are packaged into a fascicle. So an individual axon is going to be covered by a sheath called the endoneurium, and so the individual muscle cell is going to be covered by a sheath called the endomysium. So very similar prefixes to point out there in the anatomy of muscles and nerves. Epimysium, paramecium, endomysium covering the cell. Now, when we did the neuron here, what you got beyond this was the endomysium is surrounding the thing, and the axon itself is normally insulated with myelin. But you already know there's some exceptions to that rule, don't you? What are those? Postganglionic projections of autonomics. Yeah, unmyelinated. Okay, so that's the anatomy there. Now, whenever you cover the individual muscle cell, do the nerve first. You cover the individual neuron, you have this endoneurium around it, and the myelin is around the axon. What I did not say to you was the axon itself is what you expect a cell to be. In other words, it has a plasma membrane, just like any cell does. Now that will become really important for us because what proteins are in that cell membrane will dictate how the axon actually works. Same exact thing is true for the muscle. The individual muscle cell is covered by a sheath called endomysium, but within that endomysium, the muscle cell also has a plasma membrane, and I've already named it for you. It's called sarcolemma. So beneath endomysium is sarcolemma in the anatomy of the muscle. So this picture shows you the sarcolemma around an individual cell. Now, notice in the anatomy of the muscle cell, things begin to get a little more interesting now. At this point, we're diverging in the anatomy comparisons between muscles and nerves. Whenever you look in the cytoplasm, if you look inside the cytoplasm of an axon, you're going to find a microtubule network that has to be seen with, very high power microscopy. When you look inside an individual muscle cell, it does not look like a typical cell in the body at all. Instead, the theme of continuing to describe the anatomy with ever increasing numbers and decreasing in size, cylinders build the cell, build the muscle. So from the bottom up or from the top down, either way you go, the whole muscle itself appears to be a system of cylinders. Kind of like the kids' swords that you have where the little one goes inside the big one, you put it down and you pop that thing out. It's a good illustration of the anatomy, the ever increasing in number and decreasing in size number of cylinders. So this doesn't look like the inside of a cell. When students look at this, that's the sarcolemma. I'm talking about now an individual cell. There's the ba-bump I described just a moment ago. This is inside the cell. So I'm looking for things like the nucleus, the mitochondria, and the lysosomes, and the ER, and all that stuff. The prototypical cell. It's not what muscle cells look like. Now, do they have nuclei? Yeah, you learned this. They have multiple nuclei where the cardiac muscle cells are uninucleate. Remember that from way back? But the domination inside the muscle cell is protein. This is a recurring theme in anatomy and physiology. If you want to describe the anatomy, you describe the arrangement of protein. And that is exactly the way you describe the inside anatomy of a skeletal muscle. All right, so what are these cylinders? These cylinders are called myofibrils. So an individual muscle cell in its cytoplasm has cylinders in it, and those cylinders are called myofibrils. These are literally the protein content of the cytoplasm of the muscle cell. So there are a whole bunch of different proteins here, and I'm not going to give them all to you, but you have to have enough to describe the gross anatomy. So first of all, let's notice some broad description things here. Notice in this particular section of skeletal muscle, you can see the striated bands. And we call these bands dark and light bands, A bands and I bands. I will expect you to know what the proteins are that build these bands. So I'm going to teach them to you. So this now is labeled a sarcomere. But what is this really? Okay, one more time. Here's the cell. It's built out of cylinders called myofibrils. So now, what am I looking at? This is a myofibril. Okay. Now the reason it is labeled sarcomere here because this is a slide I used to teach sarcomere with. Technically, a sarcomere is the distance from one Z-disc to the next one. A sarcomere is the functional unit of a skeletal muscle. This is the core unit of the muscle cell that will be used to describe its function, the sarcomere. So really, in terms of the labeling here, this would have would be better identified as a myofibril / sarcomere. There is in fact one sarcomere sitting in the middle here, but from here to the next Z-disc would have been another one, and from here to the next Z-disc would have been another one. Okay, so let's describe the bands. First of all, the A band and I band situation is doesn't have anything to do with the function of the muscle. It's literally how the anatomists describe these pieces of tissue as they show up through the lights of the microscope. The words stand for isotropic and anisotropic, which really have no anatomical or physiological meaning. So I never write them on an exam. But you need to know at least what the A bands and I bands are made of. Okay. So first of all, notice the I band, which is normally called a light band, has a Z-disc in the middle of it. And students want to know, what is a Z-disc or a Z-line? So you can tell it has a kind of zigzag appearance to it, but all these terms that are used to describe the anatomy were described by Germans, and they all have German names for them. They're not Latin names. So I'm not going to try to pronounce all of them for you. But it really means a disc or a Z-line or a zigzag arrangement in the middle of the light band there. So it doesn't have an anatomical particular meaning. It's good for you, I think, that it's called Z here because it does look zigzag. So you can remember the Z-line. Now, you can see here, the middle of the I band where the Z-disc is, that the blue line there indicating the Z-disc is a thin line, which is appropriate because that Z-disc or Z-line you can see is attached to some very important proteins called actin proteins. They are considered to be the thin filaments of the individual muscle myofibril. So the Z-disc attached to these thin filaments, these actin filaments, and the portion of the muscle cell where the Z-disc and actin are is considered the I band. This is the lightest portion of the striations that you see in the muscle. Now, the dark striated portions of the muscle are called the A bands. And you can see there's another protein here. This thick red line here indicates the thick myosin filaments. So this is myosin protein. So the two big proteins used to describe the anatomy of an individual muscle cell, myofibril / sarcomere, are actin and myosin. So you can see the I band is actin and Z-disc. The A band is made up of actin and myosin and an H zone, which you can see labeled here. Now let's talk about that for just a minute. What's not shown here is there are a number of other proteins that are part of the anatomy here. And really, I don't think at this point it's necessary to teach you all the connections of titin and nebulin and the C protein and all the other proteins, the myomesins, all these different proteins that are connecting these things, the cytoskeletal proteins that connect them. But sometimes students will ask, and so I'm trying to teach you the gross anatomy parts here. How do you describe the gross anatomy? And you really do that with the actin and the myosin. Now, I'm going to add one more piece to this because I will use these other two proteins to talk about the function next semester. So I want you to know them. All right, so let's finish this first. So the A band is both actin and myosin, but can you see here that the actin actually runs out about halfway across the A band, which means there's a portion of the A band that does not have actin there. So it will be, for the close inspector of the muscle anatomy, this will be a lighter band. Will it be as light as the I band? No, because the myosin is a thicker protein, bulkier, so the light doesn't pass through it as easily. And so this portion in the middle is called the H zone, where we find the M line, where titin's attached. You don't need to know that. The M line is the center of the H zone, and that's a German word, Healy, I can say that one, which means the light zone. This is the light band of the A band, the center where there is no actin in the middle. So really, in terms of shading, in terms of gross anatomy on the muscle, you have a very light band that is thin filament only, Z disc and actin. Then you have a thickened portion, a darker portion here of the A band where actin and myosin overlap. And then you have a third shade, something in between the two, right in the center of the A band, called the H zone, where there is no actin. There is myosin only in that H zone. Okay, so that's the anatomy, the breakdown of the A band and the I band, and the Z disc, and the thin filaments and the thick filaments. Now, one more piece. Let's take this one step further. Notice here, as I did on the last slide, the sarcomere defines the distance here between two Z discs or Z lines. This is the functional unit. And next semester, I'll recognize the word I just said here, tighten. Next semester, that's not necessary for you. Next semester, I'm going to walk this down one more time for you very quickly, like in about 90 seconds, because you've had it in anatomy, to the point where we're talking about the interactions between the thin filaments and the thick filaments. So, can you tell me which band I'm looking at here? A band. And what is this portion of the A band? The H zone. Okay, now notice the myosin filaments. Each myosin filament has two globular heads that stick off of two little tails. And so they're all bundled together, these thick filaments, proteins. And these little globular heads have to interact with the actin in order for the muscle to contract. Now, I like this picture because it's really closer to what we would consider reality. And you can see how the myosin heads tend to be oriented. See how the myosin heads tend to be oriented like this? And that's important because these myosin globular heads, when they interact with the actin, are going to literally pull the actin filaments toward one another. They're going to slide over the myosin proteins. In the model for skeletal muscle called the sliding filament theory of muscle contraction. So that's where we're headed later in your lives, the globular heads interacting with actin. However, in order for that to happen, there are two other proteins not shown here associated with actin that you must be aware of. And so here they are. They are troponin and tropomyosin. So let's describe how they are anatomically associated with the actin filaments. First, the tropomyosin. Tropomyosin looks to be laced within the actin filaments. So there's F-actin and G-actin, and this thing really looks like a pearl necklace that has been twisted. Can you see that in the illustration? It looks like a necklace that has been twisted. The arrangement of tropomyosin in that necklace is illustrated very nicely by thinking about hair that might be tangled in a pearl necklace. So let's just say, for illustration purposes, you left your pearl necklace in the drawer with your hairbrush. And when you pull the pearl necklace out of the drawer and you twist it, you can see that there's hair tangled all in the necklace. That is a great analogy, in my opinion, for the way tropomyosin orients with actin. The difference in the analogy in real life is it's not a random arrangement. The tropomyosin actually, can you see the little darker purple places here? The tropomyosin actually is oriented on the actin filaments, on the binding sites for the myosin heads. Tropomyosin covers the binding sites for the myosin heads. That's where it is normally when the muscle's at rest. So guess what you have to do in order to get a muscle to contract? You have to cause a configuration change. There has to be something that moves the tropomyosin off of the binding site. So tropomyosin blocks the interaction because of its anatomical position. It blocks the interaction between myosin heads and actin. Okay, so that's tropomyosin. Here's the last one. There's a three-part globular protein called troponin, or the troponin complex, that also associates with actin. This particular protein, this troponin protein, has 3 different binding domains. The first binding domain of the troponin, you can see here very nicely, is associated with tropomyosin. Very important anatomical connection. Troponin is connected to tropomyosin. So I wonder who's going to be moving the tropomyosin off the binding site. This is the protein that's going to do it. It's the one attached. Now, in addition to having a binding domain for tropomyosin, troponin also binds to the actin filament. So it's hanging on to the tropomyosin, and it's hanging on to the actin. The third piece here, which there's no indication on this, but let me back up to the first slide you saw here today. The third piece of the troponin, this is the first picture we looked at right here, and it showed the role of calcium here. And what it doesn't show is the role of calcium in here, but this one, you might as well put a big asterisk by this one. When you talk about muscle contraction, and this will be true of skeletal, it will be true of cardiac, it will be true of skeletal, it will be true of smooth, it's going to be the same story basically. In order to get the thing going, in order to get it to do what it does, you're going to have calcium. And so, interestingly enough, this protein has a binding domain for calcium ions. And it is the binding of the calcium to the calcium binding domain of troponin that will ultimately allow myosin to get to its binding site on actin. Okay, so that's why I bring it up. We'll have a lot to do with that, maybe over an hour talking about how that happens. But today you have learned the anatomy. You feel good with the anatomy of a muscle. You can take it from the epimysium to the whole muscle, to the paramysium around the fascicle, to the endomysium around the cell itself. Then the cell is covered by a membrane called the endomysium is around the cell, and inside that is a membrane, a cell membrane around the cell proper called the sarcolemma. When I get inside the sarcolemma, I am constructed now out of the arrangement of proteins. They are arranged into sarcomeres that build a cylinder called a myofibril. Now, if I deconstruct the myofibril, I find A bands, I bands, and the arrangement of actin and myosin and troponin and tropomyosin. Okay, so that gets the whole anatomy of the muscle done. All right, the last thing then is bone. So let's talk about the ultrastructure the structure of an individual bone, which I've introduced to you. So this will be the second time through, but this time you got to pick up the rest of the details because I won't do this again as we progress through the dissection. Now, here's some things you know already. For bones, you already know that long bones have an epiphysis and a diaphysis. And I took a great deal, made a great deal of effort and time trying to explain to you in terms of tissues, what kinds of things would be associated with the ends of the bones and the shafts of the bones. So if I were to look inside the epiphysis of the bone, within the trabeculae, within the diploid, within the cavities of this epiphysis, what kind of tissue would I find in those cavities?
Red bone.
Red bone marrow. And what is that exactly?
Reticular.
That is reticular connective tissue, that's right. Now if I work down into the shaft of the long bone, I know that the bulk of the mass of that bone is made up of the particular bone type that we looked at histologically. Like bundles of pine trees with concentric rings. That type of bone is known as compact bone, yes? Or would you say?
Cells, osseous.
Okay, it's osseous
Cells.
tissue, but it's compact, or lamellar bone. Lamella means rings or layers, and so it's lamellar or compact bone makes up most of the shaft. But there is also spongy bone there too, right, with trabeculae, that has marrow inside, a medullary cavity, but it's not reticular. What is it? It's adipose tissue. So that is the yellow marrow that's found within the medullary cavity of the shaft. So these are things that you already knew. Now, notice on the end of this, I'm going to make a bigger deal about this today, I also pointed out to you when we talked about hyaline cartilage, I said, before we're done with the introductory material, we're going to learn the process of making a long bone. Remember that? So all the long bones and all the carpals and tarsals in your skeleton, originally they were hyaline cartilage. So when the bone has completed its development, there will in fact be a cartilage piece on the epiphysis. It's labeled on this side. That is hyaline cartilage, and we call it articular. Now, in addition to the hyaline cartilage that goes around the ends of the bone itself is covered with a membrane called the periosteum. Now, the bones we have in the lab they don't have a periosteum on them. The periosteum is in fact dense, regular connective tissue that fuses with tendons and ligaments. It is completely indistinguishable from a tendon or a ligament where there's an attachment. Now if you take the bone out, a living bone out of the body, you can see that there's a glistening membrane all the way covering the outside of the bone. But if you look at where a tendon or a ligament attaches to a bone, where the ligament comes to the bone, there is no distinguishing way of telling where the periosteum starts and ends and where the tendon or ligament starts or ends. They are fused as one piece, which means the connection from tendons and ligaments to bones is not really the periosteum. It is in fact the way the periosteum attaches to the calcified matrix, to the hard part of the bone. And so this picture shows periosteum labeled here. And if you look over here on this picture,